All right, Doctor, you can go ahead and screen. And share your screen then.
Okay.
All right, Doctor, can we go ahead and start? All right, perfect. Thank you. Hello and welcome to MedNews Week conference, where our goal is to bring you the most cutting-edge information from medicine's global leaders. I am Ahmed Hashim Aziz, and I will be your host for today's session. Before we begin, I want to first acknowledge the great team that helps make these events a reality. Our founders are two internationally recognized researchers, Drs. Jan Leifman and Chandler Park, who is also the president of Kentucky ASCO. Our chairs include Drs. Park and Leifman. Our associate directors include Madhuri Balasubramaniam, Muskan Joshi, and Gayatri Menon. Our associate managers include Alexandra Vandekleift, William Wilkerson, Pallavi Pai, and myself, Ahmed Aziz. Our educational committee includes Sean Jakovitz, Shubha Pawar, Choya Harshal, Reda Khan, Sri Valabeni, Helena Columba, Soumya Nadar, Rabab Haned, and Imad bin Zahid. We also want to thank our partners, Bumedi, who is a global leader in medical video educational content, and the National Society of High School Scholars, an honor society founded by classes of Nobel of the Nobel Prize family. The moderator for today's event is Helena Koloma, and now to introduce our great keynote speaker for today, Dr. Harpreet Singh. Dr. Singh is a global authority in precision medicine and targeted therapeutics, novel trial design, innovative regulatory initiatives designed to expedite drug approvals and use of real-world data in regulatory decision-making. She has also led or overseen notable approvals in lung cancer, including targeted therapies for MET exon 14 skipping mutations and RET fusions. She is also a dedicated advocate who engages with the greatest scientific community to increase the evidence base for treating older adults with cancer. With that, it is my pleasure to welcome Dr. Singh. The floor is yours, Doctor.
Thank you, and thank you so much for having me. I am thrilled to be here tonight, and whenever I give talks, I always get a ton of questions, so I'm hoping that we have enough time to chat at the end. I titled my talk, Innovating Clinical Trials and Patient-Centered Drug Development, and I hope that, by the end of this talk, you will see that those of us oncologists who have dedicated our careers to public service are truly dedicated to patient-centered drug development. Everything we do revolves around that. That was an excellent introduction. My official title, I am a medical oncologist. I direct one of 3 solid tumor divisions at the FDA, and I also hold a role in terms of cancer in older adults. So I'm going to talk to you a little bit about FDA's mission and an introduction to our little corner of the FDA, which is the Oncology Center of Excellence. Take you through some very interesting data, some recent trends, big data, if you will, in oncology drug development. And then I want to walk you through 2 projects, which I am helping lead with some of my junior staff. Project Site Selector, for those of you who may be going to AACR or who pay attention to kind of what we're tweeting about. This is about the concept of developing truly multi-regional clinical trials. And then Project Pragmatica, which is what we're calling kind of back to the future, actually going back to this idea of just very large and simple randomized trials. So what is the mission of the FDA? So first of all, we are responsible for protecting the public health and by ensuring safety and efficacy are the 2 main components we look at of medical drugs, in our case, for patients with cancer in the OCE. We do not take any cost or payment issues into account when we're looking at drugs. So we actually don't know how much they'll be priced for, and we have no leverage over that either. And we don't regulate what we call the practice of medicine, meaning just that because we approve a drug does not necessarily mean you have to use it. And we also don't kind of regulate sequencing and things like that. At the FDA, more so than any other workplace I've been in, more than internship and residency and even fellowship, I have never worked so much as part of an interdisciplinary scientific team as I have since joining the FDA. I work very closely every day with PhD toxicologists. They help me understand novel drugs and what they're looking like in the animal models and what toxicities I could expect in a human. And this allows us to then kind of create DLT criteria that might make sense based on the expected toxicities of the drug, or more monitoring, for example, if there's going to be ocular toxicity expected. My clinical pharmacologists help with determining the optimized dose of a drug. There's a lot of attention paid to this. There's product quality, which is kind of how a drug is manufactured, where it's being manufactured, is it in a sterile environment, are all the protocols being followed? This is part of a drug approval. It's often not thought of, but it's very critical. And there have been shutdowns of companies, production lines that have held up drugs. There's always drug shortages. And of course, our statisticians, very dedicated, some of the brightest minds. We've really developed a great expertise. And then, of course, we have our project management. And the oncology group is unique because we are specialists in our particular tumor type. So you have your breast oncologist, your lung, GU, etc. We are housed in the Oncology Center of Excellence. It is the first ever interagency institute. So it's the only institute actually so far at the FDA. I think they're thinking about forming another one. But we sit here in CDER, which is a Center for Drug Evaluation and Research, whereas, for example, the CAR T cells are regulated here in CBER because it's considered a biologic. And I am the director of this green circle division of Oncology 2. And we do, lung, head and neck, and some other tumor types as well. We've been very busy. And 2022, we saw cancer drug development continue to progress rapidly, despite the lingering effects of the COVID-19 pandemic. Some approvals are highlighted here, and I just want to mention a few of them. This one, the Lucium 177, this was a first-in-class radioligand therapeutic for adult patients with PSMA, prostate cancer, castration-resistant. You've had several new bispecific antibodies for follicular lymphoma, for multiple myeloma, for uveal melanoma. We had approved here a new antibody drug conjugate for certain gynecologic cancers. This approval of trastuzumab was a HER2-directed antibody for HER2-low breast cancer. This was really groundbreaking. In my division, we had this sodium thiosulfate. This was for children to reduce the risk of ototoxicity, hearing loss, which is associated with cisplatin. So very busy year in 2022. And we also make a lot of policy statements. That's one of the things I love about being a regulator, being at the FDA, and it's a scientific-based agency, and we develop policies around drug development. I'm going to highlight a few here. Tissue agnostic drug development. This guidance came out. They're available online. And this is basically an idea that you could have a drug that targets specific molecular alterations across multiple tumor types as defined by organ or tissue, as opposed to just one drug, one tumor type. So that we're targeting the molecular alteration that can be seen across tumor types. And of course, at the very bottom, diversity plans. This was a very crucial guidance, which basically gave very direct instructions and advice on how to work towards developing oncology drugs that enroll representative numbers of patients from racial and ethnic populations in the United States. Apologize, jumped a little. Okay. Now I want to take you through some kind of bigger trends in oncology product development, and this is an FDA analysis. This information is embargoed, meaning it cannot be shared. I know we're being live streamed, but you cannot distribute these slides. And this work was done by Emma Scott, who is a PhD, and she was in we do have an NGS lab at the FDA. So Emma presented this amazing data, and I immediately asked her for her slide. So she looked at all oncology approvals over the past 22 years. And she excluded some, and I won't take you through this, but basically think 20 plus years of approvals. Okay. And she developed a new kind of classification system by mechanism of action. And this is, and so you'll see these trends in a moment. So what's important to remember is the red is a targeted biologic. The yellow is a targeted drug. So they are different. Whereas the blue are just your classic kind of cytotoxic drugs, chemotherapy. And this is what we saw overall in our trends, is that you see this upward trend in red of targeted biologics. Similarly, in yellow, you see these targeted drugs. And, you see a flatlining here of cytotoxics. And I think that just reflects, where drug development is going. And then in terms of actual approvals, you see them going, all products here in black. Over the years, you just see this exponential increase in actual drug approvals. And, the data does show that the mean annual oncology approvals was 7.4 per year in the early 2000s. And more recently, we're approaching close to 60 a year. And most of these are targeted drugs. So great news for patients. This is just some data looking at disease site. So good news for patients with lung cancer, lymphoma, GU cancers, breast cancers, and leukemia. You see these kind of very big movements for them. And on the lung cancer issue, I wrote a paper which was published in JAMA Onc with Dr. Rick Pazder, who's my boss and mentor at the FDA. And we talked a little bit about how you do see actually now a decrease in mortality for patients with lung cancer, which is decreasing faster than the incidence of lung cancer is decreasing. So thereby kind of, alluding to the idea that the mortality is decreasing, not only because patients are perhaps not, we're not seeing as much use of tobacco, but also because the drugs are doing something. And I thought that was great data. Across product classes, you see the most approvals in this top row is kinase inhibitors. And they also had the most distinct products. And second to that are immune checkpoint inhibitors. And then innovation. So this was an interesting kind of analysis that Emma did, where she divided all the drugs into classes and subclasses, and basically, and she defines this in her manuscript, which is in process. It has not yet been fully accepted. But again, you see the most kind of movement in targeted drugs. So a little bit of this is just to set up this idea that, of the targets, there were 83 targets for 207 approvals. So what does that tell you? Probably multiple approvals for one or two targets. And you see EGFR and HER2 being the most targeted, and a little bit more about kind of the targets and how they're all connected. This at the very end, I remember when she presented this, I thought it was just very intriguing that she did this network analysis, and basically all of these target proteins, 73 of them overlapped into only two networks. And then this is just a little bit a look at single agent versus combo. So a good number of combinations. Again, mostly you see cytotoxics with targeted drugs. In terms of combination strategies, so each of these lines, so many combinations, but I like to just follow the colors. So you see this mostly red. So this is basically mostly combination strategies utilizing targeted biologics, mostly immunotherapy. All right. Now let's take a step back a second, and I want to talk about something called accelerated approval. We don't have a show of hands here. I can't see you. And so I don't know how many of you actually know about accelerated approval, but let me tell you. Accelerated approval was developed in 1992 in response to the HIV and AIDS crisis. What was happening at that time, and some of you probably were not born, and I was born, but I was too young at the time to know what was going on. But what was happening at that time is that patients were dying. They were dying of this new virus that, was yet to be characterized. And there was a desire for novel therapeutics. Okay. And people were literally on the FDA's campus protesting and demanding earlier access to drugs. Heart therapy was being developed, and patients said, you have to find a way to get us these drugs sooner than conducting a large, randomized trial with a time to event endpoint like progression-free survival or overall survival. What you can see in HIV and AIDS is that the CD4 count drops. It's a very easy measure of disease activity. And this is important to remember because I'm going to come back to this. You have a surrogate or a marker that you can use to determine early benefit. So although and so Congress, our US Congress passed this legislation for what is called accelerated approval. And so the idea is that you're balancing early access to products with still some uncertainty. And although this was born out of the HIV/AIDS crisis, today, the majority of accelerated approvals are used in oncology. And why? Okay, so the reason why is because in oncology, we have another kind of an analogous marker, if you will, predictor of response, which is actual response rate. So watching a tumor shrink on imaging for most patients is clinical benefit. And for us as oncologists is an early signal that the drug likely has an effect on a time to event endpoint. Oh, I keep doing this. I think I'm touching my keyboard. And so accelerated approval is something where we may approve a drug if we determine that the product has an effect on a surrogate endpoint that's reasonably likely to predict clinical benefit, or on an endpoint that can be measured earlier than survival. So again, in oncology, that's mostly response rate. So that's here. This is where you see kind of the rise of the single arm trial. Can we just collect, 100 or so patients with this novel drug, get a response rate? And if we can prove that this response rate confers a benefit to patients that's a greater benefit than what is currently available to them, then this would qualify for an accelerated approval, which is basically, again, there still needs to be a confirmation of benefit, usually in a secondary trial. You may also see us use this in rare cancers like the ROS1 lung cancer. A lot of the very rare mutations in lung cancer, we utilize accelerated approval. So verification of benefit after accelerated approval. So once we grant this accelerated approval, you must, the sponsor, the drug company must conduct some sort of confirmatory trial that confirms that initial impression or that initial data that there is in fact a benefit of this drug. And it could be in a different line of therapy. It could be, the classic way is to do a randomized trial. That's what we see most often. But again, in a rare situation, it may not be feasible to randomize, so we may ask for more single arm data. You can withdraw an accelerated approval. So pull the indication off the market. I don't have time to get into this in this talk, but we have done it. And it requires a great deal of effort. In oncology, we have had 181. This is actually current as of today. I asked for an update. 181 accelerated approvals granted. And of those, half have verified their clinical benefit, and then we've converted them to what we call a traditional approval or a regular approval, meaning the company is done. They no longer have to provide us any data. The drug is on the market in full use. Whereas we've also had clinical trials fail to confirm benefit. This has happened several times. We've actually had 23 indication withdrawn. So these are some of the trends that we're seeing in accelerated approval. So you see a slight decrease in the last 5 years, and it's just kind of stabilized. But again, throughout the FDA, you do see oncology most heavily utilized accelerated approval pathway because of our endpoints. And then again, going back to, of course, I think this tracks, but you see in orange, the regular or traditional approvals, in pink are accelerated approvals. And you see again that the accelerated approvals tend to be in these targeted drugs, targeted biologics, which makes sense because they can give you a response rate. So I just want to summarize Emma's work. Emma Scott. So you saw an increase in the pace of oncology drug development, mostly in targeted drugs and biologics, mostly kinase inhibitors. These molecular target proteins have various cellular functions and locations along the pathway, but they're all very interconnected. You do see about a third of these approvals being in combination products, and you do also see a heavy use of accelerated approval, but stabilizing, not necessarily increasing. All right, so I want to switch gears a little bit. This is some work that I am leading, but Dr. Lottie Ackenborough, so these are his slides, which I helped him develop. And this is a little preview for you all. These will be presented in some part at AACR in a few weeks. So we are doing a project called Project Site Selector, and it's predicated on navigating oncology drug development through what we called troubled waters. I wrote a paper in the Lancet called Bridge Over Troubled Waters. About a year ago, it was published, and it stemmed from the idea of clinical trials not looking truly multi-regional, rather they were coming from either single countries or very limited regions of the world. And I want to start this conversation with a forward-looking statement, and that is that what really we're all after is what the ICH E17, which is the International Council for Harmonization, which is comprised of several nations around the world who come together to discuss various topics like clinical trial conduct and design. And they espoused and they wrote a guidance which supersedes FDA guidance. So an ICH guidance stands as FDA guidance, that the desired clinical trial design is a multi-regional clinical trial, meaning that patients from all the regions who joined ICH, which there are many of, and I personally and we at the FDA also believe more regions should be brought into the fold, that there are enough patients enrolled in these clinical trials from around the world, multi-regions, that we can actually evaluate the consistency of treatment effect across these regions and populations, and avoid the need for what's duplication of trials and what are called bridging studies. Back in the day, certain countries would say, you can go and conduct a clinical trial wherever you choose, but we want you to conduct a dedicated trial for just patients in our country. It can be smaller, and these were called bridging studies. And we're saying maybe the ideal choice is really to just start off with a broad group, particularly when we're regulating drugs for patients in the United States. We are bound to serve them, and it's a very diverse population here in the US. And so it is our responsibility to take a look at the clinical trial data that's coming in and ensure that it does, in more than one way, reflect the patients for whom the drugs are intended, whether that's by race and ethnicity, gender, age, geography. There's many factors. And we believe that conducting a true multi-regional trial will promote earlier worldwide access to new standards of care, and also just allow the international harmonization around the world, make it easier to conduct global clinical trials. And what does that mean? So sometimes a company will come to the FDA and propose a control arm, and we'll say, well, that's not actually the standard of care control arm here in the United States, so I don't see how you could conduct this trial. And they'll say, well, this is not approved in this part of the world, this part of the world. So therefore, this makes it more challenging. So we took a look at some trends, and what we have seen following these anecdotal observations is that the pace of US enrollment, this is specific to patients with lung cancer and only goes back about 7 years, and we have not published this data. We're working to fine tune it. But nevertheless, you do see this, slightly uptick trend, which tracks with what we're seeing in our day-to-day work, is that you are seeing a decline in US enrollment in clinical trials. And again, some of this data is starting to come from either one or two, very specific regions around the world, or just, with no US patients. There's many reasons for that. But why do we actually care? So some of the regulatory considerations are, if a clinical trial data set comes from very limited parts of the world, maybe one or two regions of the world, can we actually confidently infer that clinical trial to our patients in the US? There are so many factors, intrinsic and extrinsic, known and unknown, that we really cannot account for. And as I mentioned earlier, the issue with the clinical trial conduct and sometimes outdated comparator arms. And this actually is really what kind of, triggered a lot of these discussions. And what I've learned as part of this is that, that we do not live in a vacuum. Medicine is not practiced in a vacuum. We are not oncologists in a vacuum. These, global events like COVID, like regional conflicts and wars, if clinical trials are being conducted in these parts of the world, we also have to go in and inspect and, confirm or verify that data, particularly if there is minimal experience dealing with clinical trial data from that part of the world. And due to these kind of these global conflicts, or troubled waters, as we called the session, we actually cannot go in. We cannot send our inspectors safely. There's federal laws which prevent them from entering the country, whether it's our laws or that particular country's, or again, in the example of regional conflicts and wars, it's actually, could be not safe for our investigators to go in. And so this actually has had impact on drug development. I have a few applications in-house, which were conducted in one single country. However, the indication was rare and it was appropriate, for that particular population. And I cannot approve these drugs because the FDA staff has not been able to go in and inspect. So the goal of Project Site Selector is to understand how countries and in turn clinical trial sites are selected. What do companies think about when they do this? What are the challenges with US patient enrollment? Why do you see these numbers tracking down? And to really get out the idea that what we are after is truly early consideration of global clinical trial sites to make these trials truly multi-regional. Okay, now I'm going to transition now and move on to another project, which again, I do not do any of these solely, but these two that I'm choosing to speak about today, I chose because they're closest to my work and I do, I am playing a leading role in them. So Project Pragmatica. So this I work with Dr. Donna Rivera. She is a PhD bioepidemiologist, and she handles all of our work around external controls, real-world data, and pragmatic trials. The goal of Project Pragmatica is to innovate, explore trial designs that make clinical trials more functional, more efficient, and always patient-centric. I love this slide because I think it makes it very easy to understand what is the difference between a pragmatic trial and a traditional randomized clinical trial. Well, in a pragmatic trial, the idea about the design is that rather than have the patient come for additional visits outside of routine clinical care, that those visits are built into routine workflow. And that as a result of this design, you would have a more generalizable population because it's easier to participate on the trial. More sites can participate, more investigators. very broad inclusion criteria. That the data would be collected not only through, CRFs or, case report forms, but through just your electronic health record or other real world data sources. The issue with sample size is because there's a lot more variability, you don't have this very tightly controlled experiment. So you're probably going to need to up that sample size and have an easily measurable endpoint in order to kind of drown out some of the noise that you would get given all this heterogeneity. So this is relevant in lung cancer. And I received an email today, actually about an hour ago. I'm very excited to hear that Project Pragmatica Lung, which is the first of these pragmatic trials, has started enrolling. So I'm going to take you back to this past summer of 2022, when I, as the division director, received a request from Merck, the makers of pembrolizumab, for a breakthrough therapy designation. I didn't explain what that was, but it's essentially it's a designation which can be bestowed upon a drug or a combination of drugs if they are able to show an early clinical evidence that they're substantially better than available therapy. So it's just a designation, but it does buy the sponsor more interaction with the FDA, and it allows for things like if you were going to inspect, having breakthrough status may bring you higher up on the list. If a breakthrough designated drug is eventually filed for consideration for approval, that would also buy them some consideration for our timelines with which we would review. So for those of you who know a little bit about lung cancer, you know that the first line therapy is usually immunotherapy plus chemotherapy. If you look at NCCN guidelines, their category A, category 1 recommendation is pembrolizumab plus chemotherapy, whether it's squamous or non-squamous. This was based on Keynote 189 and 407. They were just the first to market. They were the first, to have this combination approved based on an overall survival benefit. And the issue is, after patients progress on this regimen, which many of them, of course, unfortunately will, there actually are no, novel approved therapies. Certainly patients could go on to get single agent docetaxel or docetaxel plus ramucirumab. It's also been approved in this space many years ago. So what Merck did in conjunction, they provided pembrolizumab to LungMap, which is a large platform trial run by cooperative groups. So this is very important. And they ran a trial, a small trial of, and I'll show this to you, but these are the top line results. We obviously granted breakthrough status. This is for patients who have non-small cell lung cancer, were treated with a checkpoint inhibitor and chemotherapy, then progressed, and they came on to this small trial. And I mean, I'm going to just kind of hit you with the results before I show you the study design, but there was a small survival benefit here. But as I'm going to show you, and again here, patients, this is a small trial, so 144 patients randomized. That's very small for the population of patients with lung cancer, which is, massive. But they were randomized to either receive investigator's choice, which I love. And what you saw here was patients could either get, I think most commonly you would see, single agent chemotherapy or adding ramucirumab, which is a VEGF inhibitor. And they were randomized. And so the investigational arm was pembrolizumab plus ramucirumab. So PD-1 plus the VEGF, and the primary endpoint here was survival. Again, here are your curves. So you see this, this small survival benefit, but I think meaningful. But the most important issue here is that the clinical trial results, because this was a small trial, it was investigational, it was exploratory, it was not for registrational intent, was not adequately statistically powered. And I think something interesting here is if you look down at the median progression-free survival and the response rate, you don't see much difference here. You don't see a benefit in progression-free survival, so that does not track. And also you see that the response rate for the standard of care arm is actually higher. So many of these factors, although the duration of response in the pembrolizumab ramucirumab arm was quite long and longer than the duration here of standard of care. But very interesting, and just we said we need to do a bigger, larger trial to confirm this benefit. I show you the safety results only to say we know a lot about this regimen. We know a lot about, and, not necessarily a worse safety profile here, more SAEs. And so we got together at the FDA and we said, what really needs to happen is a large pragmatic trial needs to be conducted with a survival endpoint, but a large, simple trial that people can get behind and that will basically hopefully confirm the survival benefits seen in these small trials. And to incentivize the companies and investigators to do this, we thought, what if we could kind of put our money where our mouth is? We've been talking about pragmatic trials for a long time. What if we could actually do this to confirm the signal of these drugs that we already know a lot about? So pragmatic trials are just one design, and but I love this. It actually is very much a spectrum. Traditional clinical trials being here on the far left. This is what we typically see, where you see them as interventional. You randomize, and there's very strict eligibility criteria. Whereas here, where we are on the third, column, is this pragmatic trial. Notice that this is still prospective, and the most important piece to this is that we are still randomizing. Unlike observational studies, or what we traditionally think of when we think of real-world data, which is just kind of a collection of data, it's most often retrospective, non-interventional, non-randomized. You're just looking at data that's already there, and that confers many challenges when you're trying to make regulatory choices. So this is the design of Project Pragmatic, of the pragmatic trial. It's actually quite a large study in which it's the same schema. Patients are randomized 1-to-1 to either get investigator's choice, and we thought that was important. And again, the same comparator arm. The protocol is very scant. It's maybe a 10-page protocol. The eligibility criteria are highly inclusive. We actually removed, or we asked the company to remove any mention of comorbidities, baseline labs. We kind of said it is in the hands of the investigator, of the clinician, of the healthcare provider. If they feel that this patient is eligible to be enrolled on this trial, they know the drugs, then let them enroll the patient. And there's also, we're only collecting very high level safety information, again, because we know both of these drugs very well. So this is a summary of this. Really, it's a proof of concept study. It is just opening in the last few weeks. It is really the ultimate lesson, I think. I'm still learning about it. It was an iterative process with multiple stakeholders, the companies who manufacture both drugs. There's two companies here, Merck and Eli Lilly. The cooperative groups who are running the trial, SWOG, the FDA, of course, the NCI. And there is a perceived regulatory risk. Companies are quite worried that we are so used to collecting so much data. Are you sure, FDA, that you don't need us to collect more? Are you going to be okay with this very limited safety data? And we at this time we are saying yes, given this very unique circumstance. Okay, so I am running up on time. I want to make sure we have time to chat. What I wanted to do is run you through some of the many programs we have at the FDA that are led by others. I spent time today talking about two, again, that I'm very close to, but we have tons of others that are really all excellent. So Project Facilitate, this is if you need help submitting a single patient IND or expanded access. This is also known as compassionate use of drugs. You call this phone number, you email this email address, and you can talk to someone on the Project Facilitate team, and they will help you get all the information in. It's been a wonderful resource, and they show a lot of great metrics. We have Project Socrates, which is run by Dr. Jennifer Gao, and she runs a series of, it's all about education. And so she has the education program through ASCO, ASH, AACR. She does a curriculum for in-house new staff. So that education piece is really valuable. Project Orbis, which was, really groundbreaking, was the very first, we had been talking to other, basically our counterpart. So Health Canada is Canada's FDA, or, Australia's TGA, etc. But we had been talking to them since 2004 about various products and so forth, and just having a collaboration with them. But in 2019, we decided to work more, it was Dr. Pazer's idea to actually do approvals with them, because we are so well resourced, or review. We don't always actually come to the same decisions. But our FDA is very well resourced, and we often move very quickly, as you know, and not all countries have those resources. And so we wanted to just expand our collaboration. We've brought so many countries into the fold. We're working with Brazil, with Japan. It's just incredibly, Singapore, we're traveling to Swissmedic, and it's been really exciting work. And I do think it's helping shift how quickly the standard of care changes around the world. I think it's happening more quickly as a result of Project Orbis. Of course, we have our real world evidence program. I mentioned Donna Rivera earlier and Dr. Paul Klutz. They basically are leading, and they have representatives from every division, including mine, but they're leading the effort to, decide how can we appropriately use real world evidence in oncology drug development. Project Optimus, very interesting. We want to work away from how we develop drugs dose in the cytotoxic era. So in the targeted therapy era, how we approach dose is different. This is a huge shift in paradigm, and we want companies and investigators to do groundwork early, not just to go up to the maximum tolerated dose, MTD, but actually what is an optimal dose? How can we get a balance of efficacy and safety? And that is sometimes a matter of opinion. So it leads to some different conversations. Project Patient Voice, this is a pilot project, but basically it allows for patient collected data, so patient reported outcomes data. If it did not meet the statistical rigor to be included in a drug label, it can be put here on a publicly available website. The companies must volunteer this, but many of them are happy to do it. And here we just put, the drug and the patient reported symptoms so that patients can go see for themselves. Project Community, this is led by by Rhea Blakey here in the, I think, third to the right, and several, Dr. Lola Fashionajay, Jamie Brewer. So this is all about, equity and really building out our presence in the greater community, maybe in spaces where we weren't always so present. Project Silver, this is actually my baby. And this is to improve the evidence base for treating older adults with cancer. It's about policy, it's about advocacy and research, and really proud of the work we've been doing. Project Equity is similar to Project Silver. It's led by Lola Fashionajay, and this focuses on race and ethnicity as it relates to patient-focused drug development. So I'm going to keep us on time. I want to hear from you. We know that oncology drug development continues to be innovative, and we're still seeing growth in this space, mostly in the targeted space. Project Site Selector, very topical, and it's really aiming to drive us towards a truly global multi-regional clinical trial. And Project Pragmatica is a little bit of a divergence, I think. It's this idea that can we go back to these large, very simple trials, but the drugs are important and the endpoint's important. Or overall survival is very easy to measure. And if you are going to capture less data, then you need to know a lot about the drugs. And I hope that I gave you just a little bit of flavor for some of our OCE projects. All of them have patients in mind and all are wide ranging. And certainly I know that some of you in the audience have not yet, decided where you think you may want to be or end up. And I can tell you that I joined FDA right out of fellowship at the National Cancer Institute. I never looked back. For me, it has been exceedingly rewarding, whether it's, in being able to continue to see patients in clinic, having a hand in policy. I've stood on podiums at ASCO, written papers. I'm actually trying to draft a paper now for New England. I'm hoping it gets accepted. So, it's been an amazing ride for me. And I think we have a lot of success stories here at the FDA with our oncologists. And of course, I want to acknowledge so many of my staff. I tried to name them all, so I didn't do a big acknowledgement slide, but I really want to give some credence to my mentor, and certainly he's my boss, Dr. Richard Pasder, who he is a regulator. He is a trained clinician, investigational clinical trialist, and also a true patient advocate. So I attribute a lot of my success to his mentorship as well. So with that, I will end and I will thank you and I will take any of your questions.
Thank you so much, Doctor, for that wonderful and insightful presentation. I think it's really interesting and really important that we were here for this presentation, especially to see the different types of clinical innovations on trials by the FDA's perspective. So to move on with the keynote session, I'd like to pass on the mic to Helena for the Q&A session.
Thank you for coming and welcome to Discussions with Global Leaders. Again, I'm Helena Coloma and we're so excited to be speaking with Dr. Singh, Distinguished Director of the Division of Oncology at the U.S. Food and Drug Administration. We received a lot of questions, so we'll do our best to get through all of them in the time that we have for you. For the first question, I'm going to be passing the mic back to Dr. Yan.
Thank you so much for that enlightening and insightful presentation. Truly appreciate you giving us a holistic overview of the FDA and everything that they do. Thank you so much to you and to the rest of your staff for the great work that you do and for helping to lead us through the pandemic as well. So a question that I had, and you briefly touched upon this, is about promising therapies like cellular-based therapies and endosomal vesicles, which recent studies and trials have proven that they have demonstrated some promise. How does the FDA assess living versus non-living drugs? If you can briefly give us a brief overview. Thank you.
Well, that's not quite my area of expertise. So do you mean like CAR Ts on the shelf versus like engineered
Yes.
Yes. Okay. Well, interestingly, I did do a detail in CBER where they regulate the CAR T cells, because I was actually very interested in what's going on with CAR T cell therapy for solid tumors, because of course, as you know, most of the progress has been made in liquid tumors or in the hematologic spaces. I don't think I can answer that question in a current framework. I mean, I can tell you that at that time, which is a few years ago, when I was in CBER detailing, there was a great enthusiasm for the development of off-the-shelf products, so to speak. And I think we will get there. It just, are they as efficacious? I think living versus, what was the term used, non-living? I think we view them, each with different risks and benefits. Really in CBER, it's very interesting that you bring this up, because whereas in CDER, the clinical staff really leads, and I think it's predicated on our existing knowledge of the drugs. In CBER, it's actually led by the CMC folks, which are the manufacturing group, so not the MDs, because the drugs are so complicated to actually manufacture that they actually tell us, what is and was not acceptable. So high level question, thank you for it. I can get you some answers, but I don't think I'm equipped to answer that.
Thank you so much. Appreciate it.
Next, over to Ahmed.
Thank you, Helena. All right, Doctor, my question is, what are you most excited about in the future of oncology diagnostics or therapeutics?
Oh, okay. Diagnostics and therapeutics, or both? For therapeutics, I think I am excited to see the next innovation. I think that we in our generation, I'll say mine, I have been very fortunate, very blessed to see really innovative drugs in immunotherapy. I remember being a fellow and just joining the group that had the first PD-1, drug, which at that time was EMD Serono's drug, which is now called avelumab, but at that time it had no name. And I knew that, I needed to see immunotherapy. But I think we've learned some lessons about this. I mean, you saw a plethora of these drugs being developed, and I think we've maximized their benefit. We've tried them in every tumor type. Certainly the targeted therapies are promising, but I'm actually seeing a little bit of a downtrend in their efficacy. So for example, in the KRAS space, I was so thrilled to oversee the development and the approval of sotorasib, which was the first drug approved that targeted KRAS patients with lung cancer. This is 13%. But these drugs are first generation, and when we approved the drug on a single arm trial based on a response rate, and then they did a randomized trial against docetaxel, which is a single agent chemotherapy. It's not that great. And there was a marginal benefit. It's called Codebreak 200 was the study. It's been publicly reported. Very marginal benefit in progression-free survival, and there's not seemed to be a survival advantage. The survival seems to be the same across the two arms. And I was disappointed, I think we all were, to see that. But we should not be shocked, because KRAS was called the undruggable target for many, many years, and this is just the first generation. What I'm worried about is that, we're going to have 4 or 5 first generation KRAS drugs come through, just for market share competition, without really somebody going back into the lab and saying, okay, let's get the second generation drug out there. So I'm excited to see, number one, the next big thing. What is it going to be? Is it going to be the TIGITs or the lag, or and I'm not seeing that yet from where I sit. And I think if anyone would see it, it probably would be me. I think the ADCs are very promising. So the antibody drug conjugates are very interesting. So that would probably be the space I would watch. And I think that these new targeted drugs we need to get our second and third generation targets. I look forward to basically every patient, being able to have a personalized approach to their cancer.
All right.
So I think in your lifetime, you'll probably see that, I hope.
Hope so too. Thank you so much, Doctor, for that answer. I'd like to pass the mic back to Helena.
Thank you. Next, over to Gayatri.
Thank you so much, Dr. Singh, for that very wonderful talk. I think we can all agree that we learned a lot today. So my question is, could you maybe speak a little bit about FDA's stance on decentralized clinical trials, considering a lot of clinical trial sponsors don't really understand how they fit within today's regulatory landscape?
Yeah, that's a great question, and I just didn't have time to get to it. COVID taught us a lot about this, and the FDA is very supportive of decentralized trials. And it's very interesting because we've always said, we have to make it easier for patients to be able to participate on trials. And we've fallen into this kind of trap when you're in industry, and I've never worked, for a drug company, but there's nothing to say that, I wouldn't. I've realized that there's a role for every player here in drug development. It's all about minimizing risk. So when you have a decentralized trial, you are introducing a great deal of risk, because you are, basically decentralizing the trial. You're kind of throwing things out. I mean, I think there's this perception that you're saying, okay, go get your labs at, where your local lab Quest. You don't have to come to NYU or, wherever the trial is being conducted. Go get your CT scan wherever you choose. We're going to send a nurse to your house. We're going to do a televisit. But what FDA is saying is that, look, there are situations where this is completely appropriate and the better thing to do for patients and feasible. And there is a decentralized trials guidance out, which explains exactly what to do, how to decentralize your trial, how to do things like a remote consent. So we're very supportive of decentralized trials. I think that the situation has to fit. And certainly, we would want the companies to talk to us first about this. And there still are challenges, let me say. I mean, there's a lot of forms that investigators have to fill out, and FDA, we are governed by laws, and some of them we cannot change immediately. So we have work to do. There's a whole session on decentralized trials at AACR that my colleagues are doing. Paul Klutz is chairing.
Thank you so much, Doctor. I think with the pandemic being how it was, I just read upon how the overall benefit of decentralized trials would be and how it could have like long-term implications as well. So thank you so much for that answer. Back to you, Helena.
Thank you. Next question over to Sean.
Thank you, Dr. Singh, for that incredible talk. The pragmatic trials are very interesting to me as someone who wants to go into oncology. So I'll be really looking out for those results. My question specifically is about, with the improvement on liquid biopsies for diagnostics and for personalized tumor tracking systems like Natera offers or Tempus, how do you think clinical guidelines will change for, diagnosing and then personalized?
No, I think you're so, well, first of all, I'm glad to hear you want to be an oncologist. It's a great field to go into. I think the best, obviously. ctDNA, circulating tumor DNA, is a big area of focus for us, and I want to use it. And I have seen several clinical trials in lung cancer which are utilizing it. I do think that there's some work to be done in validating this as an approval endpoint, which is, of course, a goal of many. And we just need to see more data. So, does the tumor shedding detection of ctDNA, is it uniform across tumor type? So do you see this in lung and breast same as you would in bladder? Is it different based on the mechanism of therapy, whether it's a targeted drug or a cytotoxic? Do the time points matter relative to when you administer the therapy? Obviously, you could, obtain a ctDNA diagnosis. But then how would you actually monitor them for, improvement? Would it track with imaging? So where we are right now is in the data gathering phase. And there's a lot of data out there. There's actually a project called CT Monitor, in which a bunch of companies, and I think this is an excellent example of drug companies working together, because you don't see that much. But basically providing all their data to a central organization, which in this case is Friends of Cancer Research, which is what a great advocacy group for us and for patients with cancer and for FDA and our efforts. And they're looking at this kind of across, it started off in lung, but they're looking across multiple tumor types and multiple stages of disease. I don't think it's in the short-term future, like 1 to 2 years, that you would see ctDNA used to make treatment decisions in clinical practice. I think it would only be right now in the context of a clinical trial. I think that's appropriate. But I do think I think it's one of those things that in our lifetime, in our career, definitely yours and hopefully mine, we will see ctDNA being used to make treatment decisions, to escalate therapy, to de-escalate therapy. I do believe that. However, let me just say this. Right now, I am hearing that if a physician, outside of the context of a clinical trial, pulls someone's ctDNA out of interest, or to help make a decision, the patients it's very costly. I mean, the technology is very costly, so they could get a bill for like $3,000. And I was told this directly at a conference recently. So we have some ways to go. Hope maybe one of you in the audience can develop some more facile technology that would bring costs down.
Thank you very much. Yeah, I'm hoping to see it, really change the field of medicine. Hopefully soon. Hopefully very soon.
In the interest of time, Gabrielle will be asking our last question. So over to you, Gabrielle.
Hello everyone. Hello, Dr. Singh. Thank you so much for being here with us and for your clarifying presentation. And about the accelerated approval process, it's really hopeful news as well. And I have one more personal question for you. I would like to know, what is one of your biggest career challenges and how did you overcome it?
Oh, that's interesting. Okay. Okay, that's a great question. So as you all see from my title, I am a director of a group of about 20 or so medical oncologists. And I, interviewed for this position from internally and obviously was promoted, and somewhat with perhaps not the longevity of experience that some of my counterparts had. And so I knew that I, was ready for the work, but I think that you will all learn in your lives that management of human assets or human capital or management of people is something that all the medical school and all the residency and training, it is a skill that you really have to develop. And my greatest challenge has been this role of director. I took on the position. I joined the FDA in 2015. I became director at the end of 2019. So a little over 4 years at the agency. And my greatest challenge in being director is not necessarily the decision making, because I am very decisive. And some things are very complex and they require a lot of interagency discussion. So it's not always my decision. So one challenge is really kind of managing from above and from my staff. So kind of playing, in the middle. So that's one. But my biggest honor really is also my biggest challenge. And that is to develop my staff just as I have been developed. So I find it just very intriguing and very rewarding to mentor. So I find out what makes my staff tick. What is it that you love? Do you love giving podium presentations? Do you love writing? Do you like advocating for patients? Do you want to go out into the community and meet patients and patient advocates? And then I really work to chart out a path to keep these oncologists, keep their careers developing and growing. We have, goals. What is your goal for this year? Do you want to submit an ASCO abstract? Do you want a paper in JCO? How can we kind of get you to a career goal that fits with your life? If I could say one thing, it's just that we are more than physicians. We are whole people. And so really fostering and developing whole people who are satisfied and as fulfilled with their careers as I am is an honor and a challenge.
Thank you very much, Dr. Singh, and we really appreciate you for being here with us.
Thank you so much for having me. This is a lot of fun.
A huge thank you to our speaker for an amazing session today. Was there anything that you would like to say?
No, I really appreciate you inviting me, and send me any feedback if you'd like.
Thank you very much. And thank you again to all of the great questions and to all of our attendees. Please continue to follow MedNews Week on all of our platforms and be sure to catch the replay.